Welcome and thank you everyone for tuning in to this event hosted by the World Economic Forum on the ocean twilight zone, part of the ocean that is relatively undiscovered, but you'll hear a lot more about it today. Quick housekeeping, we'll keep the presentations reasonably short, leave plenty of time for question and answers. If you have a question, just type it in the Q&A box below and we'll get that answered as soon as we can. So, Catherine, if you want to roll the slide deck, let's get started. So the project that we're going to be talking about is based at the Woods Hole Oceanographic Institution in Woods Hole, Massachusetts, on the east coast of the United States. Woods Hole Oceanographic Institution was founded in 1930. It's the largest independent oceanographic institution in the world, and it is well known and respected for not only great science, but great technology as well, really cutting edge, both science and technology, and that in some sense you'll hear a good amount today. The program that we're going to be talking about is the Ocean Twilight Zone, and you're going to hear from several of the scientists involved in this project. I should say that it's definitely not everyone involved in the project, so there's certainly will be other people who are involved, and by all means check out the website to learn, if you'd like to learn the full breadth of the project. We can only touch on a few things here today. We can go to the next slide. So what is the Ocean Twilight Zone? It's this part of the ocean, the open ocean, between about 200 meters, so 600 feet, and 1,000 meters. And it's a relatively unknown. It's deep, it's dark, it's cold, but it holds what turns out to be an amazing amount of life. And part of that life actually migrates every day up from depths during the day to the night, to surface waters at night, forming actually the largest animal migration on the planet every day. And this migration forms a huge wave that travels across the entire global ocean every 24 hours. So there's lots of really interesting, unique things about the Ocean Twilight Zone. And this project is uniquely designed to do that. The funding that we have for this project is relatively interesting for us. It's funded through the Audacious project, which is housed at TED. This is Heidi Sosik giving a talk on the main TED stage in Vancouver in 2018, outlining this project. So the Audacious project brings together private philanthropists and to fund these projects that are supposed to be obviously audacious. And we'll get to how our project, we believe, is audacious in a couple of seconds. So it's not business as usual for us. It's different. And the three pillars of this project are science, technology, and these two pillars Hui does regularly and does exceptionally well. So it's not necessarily anything new for us, the science and technology component. What is new for us in some sense is engagement, taking that science and technology and applying it to real world problems, in this case, the conservation and sustainable use of the Ocean Twilight Zone. What's new here really is that these three pillars are actually, certainly we do science and we do engagement and we do technology. What's really unique in my experience with this project is that all three of these pillars have a roughly equal emphasis. And so that's really interesting for us and has some challenges as well. But as you see, I think we're doing a pretty good job so far. So the idea then is scientific knowledge to sustainable use and conservation of ocean resources. The science themes are broad: biomass and diversity, food webs, life histories and behaviors, carbon and climate. Turns out there's really interesting links between our climate system and the Ocean Twilight Zone as well. So that's the scientific theme. But again, what's unique about this project for us is the three pillars, and particularly in some sense, the engagement pillar. So it's about more than simply doing good science, doing good technology. It's actually about bringing all that together and really making a difference. And that is what's unique about this making change happen, working with conventional print media, with social media experts, with all kinds of people that working to take that scientific knowledge to change the way that the general public and the global population thinks about the Ocean Twilight Zone, and perhaps even the ocean in general, and to really make a difference, to make that change happen. And so our first speaker today is actually really very involved in exactly this. Porter Hogan is an oceanographer emeritus at the Woods Hole Oceanographic Institution, and he's going to talk to us about the policy implications of the ocean twilight zone and how we might take that science to, in some sense, to make a real change. So Porter, I'll turn it over to you.
Great, thank you very much, Simon and KR. It's a great pleasure to have the chance to participate on this panel today, or this evening. The program at Woods Hole that's been organized to study the twilight zone has as its goal the sustainable use of the resources of this area of the deep ocean, which is to many a remote, difficult to access, and therefore a very unfamiliar environment. What does sustainability mean in this context? How can this be achieved? Fundamentally, the concept of sustainability implies an objective of fairness with respect to human well-being, both within and across generations. For the particular environment of the deep ocean, especially the twilight zone, I'd like to argue that to achieve sustainability, we need to have a way of understanding trade-offs, where we recognize explicitly that some human uses of the ocean mean that other uses might be displaced or precluded. Specifically, we can begin to talk rationally about what is gained or lost when the ocean is used in different ways. The ocean, including the twilight zone, comprises a complex array of natural resources, and I think it's important to begin to think about these resources as valuable assets, or what is known as natural capital. Natural capital can be described by its physical properties and by its value, sometimes referred to as its accounting price, when used or appreciated by humans. A central component of the value of natural capital arises from flows of ecosystem services that result from human use or appreciation of an environment, such as the deep ocean. One can think of these services as a kind of dividend. Actually, to be complete, we'd also need to be concerned with capital gains or losses, and with investments or drawdowns in natural capital stocks. But a logical place to begin is with characterizing and assessing the scale of ecosystem service flows. And the twilight zone provides some excellent examples of beneficial ecosystem services. I'll mention just briefly two specific types today and some of the trade-offs that we're beginning to explore. It's important to recognize that the conduct of marine science is essential to understanding the physical properties of the natural capital of the deep ocean, especially the dynamics of the ocean as a system and the effects of human interactions with that system. In doing so, marine science itself provides a significant source of value, what's known as a cultural ecosystem service. Please keep this in mind as you listen to the brief presentations following mine by Annette, Dana, and Simon on their cutting-edge technological approaches to understanding the ocean's twilight zone. Oh boy. Okay, KR, so next slide, please. So I just wanted to start with this typology that we've been using to categorize the different ecosystem services of the twilight zone. This typology was developed under the sponsorship of the United Nations. It's known as the Millennium Ecosystem Assessment approach. And it has been modified and refined in various ways in the last 15 years, but it's useful to start with this very general categorization. There are four separate types of ecosystem services. The first we think of is provisioning. That includes seafood, or also potentially the reduction of certain types of fish, such as twilight zone fish, into fish meal and fish oil, which then would be sold into the aquaculture industry. Also, genetic resources are a type of provisioning service. Regulating services include carbon sequestration, extremely important, as well as the assimilation of waste. Cultural ecosystem services include the conduct of marine science, and the development of an ocean literacy, or an understanding about how the ocean works and what its services are. And then finally, the fourth category is supporting services which support the other three main categories. And this includes things such as ecological links, for example, between twilight zone fish and apex predators in the surface waters, which are commercial fisheries themselves, as well as environmental condition. And next. So just a couple of examples. The first concerns commercial fish harvests and the question of whether this is really an opportunity or kind of a threat. The focus historically has been on lanternfish. There's a picture of one type on the left there, a very small fish about the size of your finger. They're the ones that undergo this diel vertical migration among other animals that Simon described. And they have been fished historically. On the map on the right, you can see locations where these fish have been attempted to be exploited commercially. It's unclear that profits actually ensued from their exploitation. And the question is, given the size of the resource, which has been estimated on the order of 10 billion metric tons of fish, 10 times more than the fish that exist in the surface waters, and 100 times more than what we remove from the surface waters every year. The question is whether these fish could be exploited commercially, likely reduced into fish meal and fish oil for aquaculture, and what the benefits would be from that. If these fish were exploited commercially, there also are questions, and in red underneath the picture on the left, what are the things that we need to think about when we exploit them? What are the potential external effects of their exploitation, including the impacts on their important predators, such as tunas, billfish, sharks, and marine mammals? A second important ecosystem service concerns this idea of a regulating service of ocean carbon sequestration. And we have to ask the question, what are the implications of climate change on that particular service? So on the right, you see the picture of the diel vertical migration again. And the animals, including the fish, play a critical role in cycling of carbon, removing it from the phytoplankton, remove it from the atmosphere, and the animals help to move it down further in the ocean for long periods of time. It's been estimated about 2 to 6 billion metric tons of carbon is moved down into the deeper ocean every year. And this provides an enormous benefit to humans in terms of avoided future damages from climate change on the order of $300 to $900 billion each year. And an important question here is, what are the implications of continued climate changes, which is acting to warm the surface waters, stratify the surface waters, deoxygenate the waters, as well as acidification in the long run? What are the implications of that to this very valuable ecosystem service? And then I'll just finish up with a final slide. We've examined a number of these ecosystem services and some of the trade-offs that are involved in a recent report called Value Beyond View. And you can go to the link on the left to get a copy of the report, or send me an email. Happy to connect you to the report and discuss this further. So thank you very much. And I'd like to introduce Annette Govindarajan, who will talk about her work on environmental DNA and understanding the biological diversity of the ocean twilight zone.
Thanks, Porter. So as Porter said, I'm Annette Govindarajan. I'm a biologist at the Woods Hole Oceanographic Institution, and I'm using molecular approaches for characterizing the biodiversity of marine animals in the ocean twilight zone. So our ocean twilight zone theme for the biodiversity theme is seeking to address several research questions, including what kind of animals are living in the ocean twilight zone, and what are the animals that are comprising this region's vast biomass? How are these animals distributed in time and space? And what are the factors that are controlling their distributions? we are using these results to inform our other theme areas in our projects, such as looking at the ecosystem functioning and biomass, and looking at how these different types of animals play into those different processes. Next. So the focus for this biodiversity assessment is utilizing genetic or DNA-based approaches to describing the biodiversity. And so these genetic approaches hinge on a concept called DNA barcoding. And so what DNA barcoding is when you have a stretch of DNA sequence in an individual animal, okay? And for animals of a given species, that's DNA sequence is very similar or the same for all of those individuals within a species. But between species, it's different. So it can be used, this barcode can be used to match species to their identified names. Okay, and so the DNA barcoding approach has been going on for a couple of decades. Traditionally, what happens is you take an animal, like a jellyfish here, you extract the DNA from that animal and you sequence it, you get your barcode sequence, you match it to the database, you put a name on it, okay? Now, some advances in the DNA sequencing technology leading to what we call the high throughput sequencing or next generation sequencing technology are allowing for the sequencing of bulk environmental samples as opposed to just individual sequences. So now what we can do is we can take bulk whole community samples. So we can, if we go to sea and we take our sampling net and we collect hundreds or thousands of animals in that net, we now grind all those up and extract the DNA from that bulk sample, and we can sequence and get hundreds of thousands and millions of DNA sequences from those samples. And so we can now very efficiently identify the types of animals in the entire community. We're taking that one step further, okay, and using the eDNA or environmental DNA approach, as it's called. And so what eDNA is, it refers to the DNA traces that are left behind by animals that were in the area. So animals shed their DNA in the way that, we shed cells from spawning, from defecation, from a variety of different mechanisms. Animals leave behind their DNA fingerprints. So you actually think of it as a type of forensics, okay? So we then take a sample of water. We don't even need the animal because their traces of DNA are in the water. We take that water, can we go back? And we take that water and we extract the DNA from the water, and then we do our metabarcoding and we get our hundreds of thousands or millions of sequences and characterize the community from that. So the exciting part of this is that we don't even need to sample the animals. We're looking for their DNA fingerprints. Next. So this slide shows this as a schematic of how this eDNA concept works. So we're sampling in the ocean twilight zone. So that means we get on the ship, we go to sea, and now we have to collect our water samples. This shows a picture of what we call Niskin bottles, which are water bottles that we can trigger to take samples at certain depths. We take our water samples, okay, we bring them back up onto the ship and we filter them. We extract the DNA from that water that was filtered, sequence it, and then we, since we have so many DNA sequences, we have to use a more complicated informatics pipeline that can give us the relative proportions and different types of those many, hundreds of thousands and millions of sequences. Next. So, so now if we want to take our samples, we, given how vast this environment is, how can we know where to take our samples? And so we use other types of information to guide our sampling decisions. And specifically, we're using acoustics, which can give us indications of where the location of certain types of biomass is present in the water column. So we can say, looking at these acoustic traces on the slide here, we see where in certain layers we might find certain types of animals, and we can use that to guide where we take our samples. Next. So we are taking this sampling approach further as well. So in addition to the biotechnology advances, what we're doing is on the sampling end, making some developments in this direction too. So if you can go back, what we're to the previous slide. And so what we're doing is using autonomous water samplers that we put on other types of instrumentation. For example, we have a newly developed instrument called Deep Sea, developed by Dr. Andoni Lavery at HUI. This is a towed broadband acoustic and imaging instrument. We can put an eDNA sampler on that and sample in response to these other types of sensed information. We also have a new robot that's been recently developed called Mesobot, and that does a lot of imaging and tracking of animals. And we have a DNA sampler that can go on that as well. So now next slide. So what we want to do is put all of this together with our biodiversity information from eDNA, from our sampling, and from our different types of tools and instrumentations, and put it all together for an integrated, comprehensive picture. So we'll take our samples with our CTD, we'll have Deep Sea, we'll have Mesobot, we'll have other types of robots. There's what we call a REMIS here. And so we can get all kinds of acoustics and imaging type of information and have our biodiversity information in that context to give us a more holistic picture. And now the next speaker, Dana Yerger, is going to tell us more about the technology that we're using. Thanks.
Thank you, Annette. I'll be speaking today about some of the new emerging technologies that we've been developing at Woods Hole Oceanographic under our Ocean Twilight Zone initiative, and also some things that we've been working on previously. So next slide. Of course, we have tools now, and we take full advantage of the existing suite of tools that we have. Tools like nets and towed camera systems and bioacoustics. But our push under the Ocean Twilight Zone project has been to supplement these traditional methods with some very exciting new technologies that can make unique new observations and take new kinds of samples, but can also take advantage of these existing capabilities and, in fact, in many cases, link them together. Next slide, please. Here's some examples. Annette already showed you the Deep Sea and the Mesobot. And we're developing some low-cost floats for carbon flux called Minions. We're making new generations of eDNA samplers, and we're pushing forward with more sophisticated navigated fish tags as two examples. I'm going to talk a little bit more about the Mesobot right now. So next slide, please. So the Mesobot is a new robot. It's specifically designed for mesopelagic studies. It's a collaborative effort between all the institutions that you see here: Woods Hole, Stanford, MBARI, and the University of Texas, Rio Grande Valley. And we've got sponsorship from the National Science Foundation in addition to the Ocean Twilight Zone. Next slide, please. Without going into too much detail, it's a hovering vehicle that can be operated as a remotely operated vehicle or as an autonomous vehicle. It has stereo imaging cameras and scientific imaging cameras. It can carry samplers, for example, the eDNA sampler, and it's designed to be minimally disruptive to the environment. For example, you see those big propellers there, part of its signature appearance. Those actually are very low-powered, very slow-turning propellers that don't disturb the animals we're trying to observe. That's one example. Next slide, please. Here's a typical mission. We lower the Mesobot connected with a very thin, lightweight tether through a clump weight vehicle. You see over here, we find some interesting targets. For example, some of these Twilight Zone animals that, again, we've captured them in nets. We see them with our towed imaging cameras, but we don't necessarily understand their daily lives very well. There are huge gaps in our understanding of what these animals do every day, how they live, how they interact, who eats who, all those kinds of questions. So our idea is that the Mesobot can go down. We can find an interesting animal. We can lock onto it with the Mesobot stereo cameras. We can then release the tether, and the Mesobot will then automatically follow that animal. Our goal is to do that throughout an entire 24-hour cycle. And of course, Mesobot carries many other kinds of sensors, traditional oceanographic sensors, and it can carry eDNA samplers as well. Next slide, please. Here you see from one of our at-sea operations with our MBARI and Stanford partners, you see the Mesobot going over the side. You can see our acoustic navigation where we can keep track of all these vehicles. Here you see one of our engineers. She's actually piloting the Mesobot during its teleoperated or its manually operated phase. And here you see the Mesobot doing its thing in the ocean. Next slide. So here's this is a still frame now of the main imaging camera from the Mesobot. And here we're imaging a specific jellyfish called Solmissus. It's a very common jellyfish in the Monterey Bay, which is where we're operating. KR, can we start this video? And here is a video of the Mesobot automatically tracking this animal. We can see many aspects of its behavior. We can correlate its motions to its propulsion. We can see the other animals that it's interacting with. There's a host of new kinds of scientific observations that we will get from this, especially when we refine this enough that we can have it observed for many hours up to a full day. And we learn how these animals live their full daily lives. And finally, the last slide here, this shows again the Mesobot on deck. And at the bottom here, you can see our eDNA sampler. It's able to process very high volumes of seawater through filters. We don't actually bring back the seawater, we bring back the filters. And from those, they can extract the eDNA and do the kind of sequencing that Annette Govindarajan spoke about. And here we see the Mesobot happily on the surface. After a dive, it drops weights like many of these kinds of vehicles and returns to the surface. We have lots of aids to help us find it on the surface, satellite transmitters and blinking lights and about every contraption you can imagine. We bring it back aboard and we can then examine the video, we can look at the sensor data, and we can process the eDNA samples. So with that, I will turn things over to Simon Thorold, and he'll talk to you about tracking apex predators.
Thanks, Danny. Yeah, and food webs in general, one of our themes is food webs, and both Joel Yuppies at Woods Hole Oceanographic and Ann Buckland at the University of Connecticut are heavily involved in this theme as well. Next slide, please, KR. And we're tracking these apex predators, and perhaps one of the most interesting things that we've found during this work is just how deep all these apex predators seem to go. Doesn't matter whether they're devil rays or whale sharks or makos or white sharks. Almost all the large animals in the ocean, at least the ones that we've tagged, appear to be diving or moving into the ocean twilight zone. We would like to know how dependent these animals are, because things like big-eyed tunas, swordfish are actually pretty important commercial fisheries and a very important ecosystem service provided by the ocean twilight zone could be forage for these commercially important species. But if it's in one take-home message from my part of this talk is that almost everything large in the ocean, at least fishes, takes considerable advantage of forage in the ocean twilight zone. Next slide. So we use satellite tags. We're developing new tags, but we're also using sort of current generation satellite tags. These are just two types of tags going on a blue shark. This is an ex-student in the Hui MIT joint program, Cameron Bourne, putting these tags onto a blue shark. And from these, we get positions when that dorsal fin gets above the water to communicate with the satellite. The second tag provides depth during the deployment. So in some sense, we're able to fly these animals three-dimensionally through their environment to find out not only where they're going, but what they're doing in these different locations. So if we get the next slide. And so what I'm going to show you is an animation. On the left, you'll see different contour colors. That's just, think of this as a weather map. Instead of showing atmospheric pressure, you're looking at weather in the ocean. And this is a mako shark that we tagged off Cape Cod. And as you'll see, the first thing it does is just swim straight across and hits what is actually a warm core eddy. So an ocean storm, a large eddy 200 kilometers across, and it hits that eddy, and it's obviously very interested in staying right in the center of that eddy. So this addresses the first question was with, in terms of the open ocean, in the sense that the open ocean looks pretty homogeneous to us, even if we view it from an airplane, but it's in fact very dynamic oceanographically, and we need to take into account these dynamic features when we're looking at use of these large predators in the ocean twilight zone. I think the next question that you're probably asking is, well, what are they doing in those eddies? And so that's the next slide. And so what we have here is a really great video of a short fin mako shark shot by Eric Savitsky. And on the right, KR is going to start an animation which shows you what those mako sharks are doing in that eddy. And so the obviously thing that you'll notice is that these mako sharks, while we think of them as primarily surface animals, they're really not. They're diving almost to 1,200 meters, so almost 4,000 feet down at various times and staying at depth for a long time in water that's cold, 5 or 6 degree water. And so they're clearly taking advantage of presumably forage prey in the mesopelagic. And so again, our question is, how important is this prey to mako populations and other populations as well, big-eye tunas, for instance, because that's going to be a really important component for Porter's models in terms of figuring out how how we value the ocean twilight zone and how we value the ecosystem services that twilight zone can provide. If we go to the next slide. So what's down there? You know, obviously work with Annette is trying to figure exactly this out. These are bristle mouths and fangfish here. So we know that there are plenty of fishes, and those actually produce really good echoes for the bioacousticians to look at. But there are also things like squid as well. Next slide. There's squid down there as well, actually probably more squid than fish, but by all accounts. And so there's really, Porter was mentioning the numbers, it's really a huge potential biomass that is down in this ocean twilight zone. And it's crazy when we think of it that we actually don't know within an order of magnitude how much biomass is down there. So, you know, we can get to the moon, but we really know so little in terms of quantitatively determining what life there is in the ocean twilight zone, and certainly how important that life might be, not only to animals in the ocean, but potentially to humans as well in terms of climate change. Next slide. So just to wrap up a little bit, again, I think we'd just like to stress that although you know, there is incredible cutting edge science and technology going on in this project, I think we will have failed in some sense if that's all we do. We're really interested in engaging the public through all kinds of traditional print media, social media, all kinds of different ways of helping the public understand how important the ocean twilight zone is to, in some sense, to their daily lives and why they should care. And so that's an overarching theme of the project. And so we have plenty of things you can visit on the website, look for new initiatives that we'll be launching in terms of engagement soon. Keep an eye on that. But it's about making change happen. It's about taking that science and technology and leveraging it and transitioning it into actual policy outcomes that will benefit all of humanity. And so with that, I think we'd like to close the formal part of the presentations. We would be happy to answer any questions. If you have any questions, just go to the Q&A button below, type in your question, and KR is going to make sure that that question gets to the right person. So thanks again for listening in, and we're ready for your questions just as soon as you type them in.
Well, we'll start with this first question from Lisa Levin. There is an effort to protect 30% of the ocean to achieve sustainability, including the water column. Has your program considered how to use the new technologies to identify areas in need of protection, or use your technologies to monitor or enhance midwater protection? This could be particularly important in areas beyond national jurisdiction and in the context of the BBNJ Treaty. Porter, you want to start off on this, and maybe Simon can chime in?
Yes, I'd be happy to. Thank you for the question, Lisa. A really important question to think about. I'm sure you know about the efforts through the Convention on Biological Diversity to establish these large scale, or to think about large scale area-based management tools, sometimes referred to as protected areas, stretching many kilometers over the ocean. And there's been a long effort to really identify the areas that need the best protection. And as I'm sure you're aware, the data supporting that efforts is heavily weighted to data that's been collected from the surface waters. And so it will be extremely important to understand the ecological linkages that Simon had mentioned and Annette had mentioned that really tie the midwater, the Twilight zone, to those surface waters, in thinking carefully about what areas should be protected. And we've been, as a team, we've been trying to track the negotiations relating to the High Seas Treaty. You mentioned the BBNJ. The long name is Conservation of Biological Diversity and Sustainable Use of Areas Beyond National Jurisdiction. And we feel that the treaty negotiations to date have not explicitly focused on this third dimension, this connection between the surface waters and also the deep seabed and the waters that lie in between. And as we've tried to present very quickly today, we think that area of the ocean is extremely important and should be used to inform management tools such as large-scale marine protected areas.
Yeah, let me follow up on that too, Lisa. The problem, in some sense, as you well know, is that the open ocean is not like a coral reef, right? Where it's relatively easy to come up with a spatial management plan that protects particular parts of the ocean. The open ocean is dynamic, and so these eddies, these fronts, these filaments, they're all moving, and so you simply can't. They're so dynamic that protecting one place and through time is not going to be as effective necessarily as protecting specific features in the ocean that we know are moving. And it's a pretty active area, more dynamic fisheries management or dynamic ocean zone management, right? Where we're thinking that we can use things like sharks and swordfish to find hotspots and find the physical oceanographic features that are linked to those hotspots. And then we can really think about how we might do true dynamic ocean conservation, and perhaps a static MPA is not actually going to be the way that we ought to be thinking about protection of the open ocean. Perhaps there are more sophisticated tools that we can use, particularly with all the technology that we have now to be able to track any kind of vessel that has the right sort of satellite transmitting equipment on it. And so I would like to think that we actually get to a point where we can start transitioning away from thinking about protecting the open ocean in static units to more dynamical conservation and management tools that are going to be much more efficient. And in some sense, they're going to provide the opportunity for win-wins, right? For more conservation and for fishermen to actually make more money by being able to catch fewer fish, spend less money on fuel, but get better prices for the fish they're getting. So I think there are some potential win-wins that we can get from a dynamic management strategy thinking as we move forward.
So our next question, I think probably Simon as well, just to speak generally on the project. Plans for Alex asked, plans for deep sea mining include release of sediment-laden wastewater from surface barges into the water column. Most of these seem to plan release at below 1,000 meters. What are the potential impacts of such activity on the meso or bathypelagic ecosystem?
It's a really good question. Yeah, so the first thing I would say, I guess, is that there's nothing magical about 1,000 meters, right? The fact that they're going to be potentially releasing those sediment plumes below 1,000 meters doesn't suddenly make them good for the ocean, or better even than releasing them in shallower water. So there's nothing magical about it. I think there are much more important things to think about where you might release that sediment than the depth of the water that you're in. I think I'm not sure anyone else is welcome to comment. I'm not sure that we really know yet what kind of damage that might do to the environment. But it's not going to be good for the environment, I guess. It's just going to be how bad, right? And I know, a lot of people are thinking about how we might even be able to monitor the environment to determine how, what's happening with these sorts of discharges if ocean zone mining, deep ocean mining, ocean floor mining actually starts happening at an industrial kind of scale.
So we have our last question. This is to Dana. Can these new technologies be also used to analyze the level of plastic pollution in remote parts of the ocean?
Well, I should say that plastic is a bit outside the domain of our specific activities in the Ocean Twilight Zone project for the moment. Certainly other investigators at Woods Hole in our larger community are very interested in those questions. Whenever we design a new kind of robotic system or a towed system like deep sea or a robot like Mesobot, we try to make it very flexible and able to carry a number of different payloads. So certainly many of the new sensors that are being developed for sensing the presence of microplastics, plastic in the ocean, could be outfitted onto those platforms and some of the other platforms that we're developing. So I think the answer is yes, and personally, I would be excited to work with a PI who had a new sensor and wanted to propose to use one of our platforms for that purpose. So we always build flexibility into our platforms. We almost always end up using them to work on a number of problems that we didn't anticipate when we first designed them. That's in fact a testimony to whether or not we did a good job of conceiving those platforms in the first place. So I guess the short answer is yes.
We had one last question. What are your plans for sharing data and observations from the program? Simon, do you want to start with this and maybe Dana can come in?
Yeah, so the project has very much embraced transparency and open access, and part of the work that's been done on this project is actually an open data platform which will make this data available and just as quickly as we can to anyone that may be interested, certainly to potentially the policy people, but everyone as well. So it's a little bit different from the way that typically we've done science, and that when we won't be waiting until the papers are published before giving people access to this data as quickly, basically as quickly as we can. Again, transparency is very important. Open access. We would like to see this data taken and used as quickly as possible, and we will be doing our best through this open data platform to make sure that it is as readily available as quickly as we can. Given the limitations that we don't want to be putting data out there that's not adequately quality controlled, but we do want to get the data out and available to people as quickly as we can.
All right, we have one more question. This is why don't you start on this one, Porter? Are you collaborating with some of the nations that have started harvesting programs? Are there roadblocks?
To my knowledge, we're not collaborating directly with nations that have harvesting programs because they have not started to harvest the fish of the Twilight Zone. We do have collaborations with researchers in Europe who are studying these questions. There are two new programs. One, the acronyms are MISO and SUMMER. And MISO especially involves the participation of the researchers in Norway. And Norway is particularly interested in the potential for Twilight Zone fish to be harvested and reduced into fish meal and fish oils for potential inputs into their aquaculture, particularly salmon industry. But there is no commercial harvesting currently. There had been commercial attempts at commercial efforts by the Soviet Union before glasnost in the 1980s and early 90s in the Southern Ocean near Antarctica. It's unclear that those efforts were commercially viable ventures, and they were discontinued in the mid-1990s. There are occasional efforts to catch Twilight Zone fish off of South Africa, the west coast of South Africa, and in the Omani Gulf, in the northern part of the Indian Ocean. Those are pretty much experimental fisheries as well. We have been seeking collaborations with other efforts that are studying the science of the Twilight Zone. And there is a recent article, I believe it was just published in Nature, that describes a collaborative effort among Twilight Zone scientific groups that are trying to understand carbon cycling in the ocean. I think that's about all I have on that. Somebody else want to join in?
Yeah, Dana, did you want to talk about other collaborations you're working on, or? Porter gave a very nice overview. I just wanted to open it up.
Yeah, well, certainly many of our activities are collaborative, and we think a lot of the technologies that we're developing and the scientific methodologies can be adopted by other groups. We can work collaboratively with them. And of course, they always, many of our outside collaborators bring their own perspective on things, which sort of drives us to use our technology in new ways. We certainly saw that previously with the new robots that we use to study the seafloor, which is what I had actually spent most of my career working on. We started off with a certain set of ideas, and I think that we very successfully pursued those ideas, but that menu, if you will, got vastly expanded when other investigators came to us and said, hey, you know, I got an idea how you can use that. And so that's sort of the essence of what we do and how we conduct our business. So I expect more of that in the future.
This is a follow-up question from Lisa about beyond MPAs. Are there many ways your very cool instrumentation could inform on conservation? Alex's question about deep sea mining and the waste of deep sea mining is alluded to one of them, but are there others that people have in mind? Porter, you've probably done the most. Yeah.
Yes, and I'm not the best person to respond to the very cool technology part of it. And Annette might be a better one, particularly with respect to how the eDNA technology can actually identify the presence of organisms in particular environments, and even discover new organisms.
Yeah, so I can chime in here. Thank you, Porter. Yeah, so I think that's true from a couple perspectives, that the genetic work we're doing with eDNA as well as with individual animals has potential to be applied to a variety of conservation questions, because in part we're establishing a baseline, and we're, in order to inform our eDNA results and to put species names on them, we are also getting reference sequences, DNA sequences for actual animals whole animals. And so we're building this reference database that then can be used beyond the life of this project for other projects into the future, so that it's a resource for not only for ourselves, but for anyone else who would be doing this kind of work, to be using these reference sequences and baseline data.
Yeah, I'll just make a couple of comments too. I think one comment might be that that I think what this project and what other projects are doing in terms of quantifying the ecosystem services provided by the Ocean Twilight Zone allows us to look at the trade-offs that we're making and the implications of those trade-offs. So, for example, is it really worthwhile scooping up mesopelagic fishes to feed them to farmed salmon if in doing so we significantly alter carbon sequestration and the movement of carbon from the surface waters into deep waters, right? So that's a trade-off that if we can actually even semi-quantify it, may not make a whole lot of sense. So I think anything that we can do to at least inform policymakers of the trade-offs that they're making in these kinds of activities, I think the better off we are. But I think the second thing is that, Lisa, you would know this better than most, but people, it's very hard to get people to care about things that they don't see any relation to their daily lives, or don't even, or find it difficult to relate to. And I think the open ocean, especially the deep ocean, is one of those things, right? We haven't got any pandas to really make an emotional connection with people that is going to want them to vote in such a way, for instance, that could lead to ocean conservation. But I think what we hope to do is to be able to link the lives of people in everywhere and across the globe with what's happening in the open ocean and in particularly the mesopelagic. So I think that's a really big challenge, and that's why we're working with people who are much better at communicating and tapping into those kinds of feelings and have the ability to reach a much wider audience that we typically do. But anything that we can do to encourage people to think about the open ocean and the Ocean Twilight Zone, I think is going to be a good thing, and it's something that we're very passionate about on this project too.
Our last question is what are your plans for observations beyond the North Atlantic? Simon, you want to take that one as well?
I can make a start. Yeah, so it was an operational decision that the cool thing about the Ocean Twilight Zone, right, is that it's throughout the global ocean. So as soon as you get off the continental shelves, you almost invariably have Twilight Zone between 200 and 1,000 meters. So it's a really interesting zone because it's contiguous across the entire global ocean. And so, you're right. In some sense, it's how easy is it to draw inferences from what's happening in the Northwest Atlantic across the entire global ocean, and of course that may not be possible. We've chosen to focus for this project on the Northwest Atlantic for logistical, among other reasons. And we're certainly going to learn things that are going to be applicable across that global ocean. But it's one reason why no one project or no one group is going to be able to do everything across the world. And that's why some of those connections, some of those collaborations that Dana talked about are so important, and Porter as well, right? It's going to take a village to do this and to do this right. And we are certainly developing tools that what Dana didn't mention or necessarily was that what we really want to do is to come up with a whole bunch of tools that are going to be relatively cheap and easy to deploy, so that they will enable pervasiveness of instrumentation that we simply haven't had up to this point. And so, you know, that again is gonna help us with those tools may be developed in the Northwest Atlantic, but if we can make them cheap, if we can make them easy to deploy, then they can be, you know, we can expand, we can ripple that model out across the global ocean to truly understand what's happening on a global level. Because, you know, a lot of the numbers that we hear in terms of the global estimates of mesopelagic fish biomass at 10 billion tons, right? It's not it's a huge number that we really need to know if it's right or not. And we're certainly not going to do it ourselves by the study in the Northwest Atlantic, but if we can develop technologies and methodologies that can be relatively easily and relatively cheaply rippled across the global ocean, then I think we will have achieved something really amazing. Dana, you might jump in by all means.
Yeah, again, elaborating on the point of, you know, more pervasive technologies. We have that thread in the ocean twilight zone. I showed one example, the so-called minion floats that can measure descending carbon flux particles, and the goal is to make them. They will cost on the order of $1,000, and they can be expendable. So that's the eventual goal. Likewise, we have plans for very low-cost eDNA samplers. So there's lowering the cost and also building things that are operationally suitable for a broader audience, ranging from our scientific colleagues all the way to citizen scientists. That's very much a goal that we'd like to do. Obviously, investigating the twilight zone involves logistical challenges that other kinds of science don't face, but we are certainly exploring those possibilities and trying to enfranchise larger groups of people to multiply our efforts by making more observations more widely distributed.
Okay, everyone. Well, I guess we're just about out of time. So I would just like to take this opportunity to thank the World Economic Forum again for hosting this event and giving us the opportunity to speak. I'd also like to acknowledge the other scientists on the Ocean Twilight Zone project who are not here today. It's a big group of scientists, and it's certainly the work that they're doing is broader than all the stuff that we were able to talk about today, but by all means visit the website to check that out, to get that in more detail. Thanks again for all the great questions, and we look forward to seeing you all soon and having some more updates from the project as we move forward. So thank you very much again for tuning in.